Hi everybody and welcome to Honors Biology at Desert Ridge High School. I'm Mr. Galladay and this is VodCast number 12. Today we're going to be talking about enzymes. Enzymes are extremely important in understanding biology. Biology, as I'm sure you're all aware, does involve an awful lot of chemistry. And enzymes are the things that make those chemical reactions happen that are in all of our cells, all of our, all of ourselves, but also all of our cells. So understanding what's going on in a cell to a very large degree is understanding what an enzyme is and what individual enzymes do. Okay, so let's get started. And the first thing to understand that this is just a little background terminology that in any chemical reaction, you have something that you start with, and we call that a reactant. And so the things that are going to react in a chemical reaction is the stuff that you start with. So that's what's typically on the left-hand side of a chemical equation. You then end up with something after the reaction takes place, which we call products. And often in chemical reactions, to make this process happen a little faster, we have something called a catalyst. Now, these are general terms, which are general terms whether you're talking about chemical reactions in cells or chemical reactions that would not involve a living thing. Okay, so catalysts, you've probably heard this term catalytic converter. Catalytic converters are something in your car which basically causes unburnt fuel to react and to burn, which causes less polluting stuff to exit through the tailpipe of your car. Okay, so an analogy of this is what happens in a marriage ceremony. In a marriage ceremony, you have two reactants which come together in the presence of a catalyst and then are changed, right? Obviously, the bride and the groom are changed by the marriage ceremony. The minister or preacher or priest or rabbi, whatever he might be, of course, is now he can go on or she can go on and do more wedding ceremonies. But those two reactants that came together, well, though they were, of course, changed by this, well, it's not a chemical, but this social reaction, I guess you could say. To put this in chemistry terms, you have a bride and a groom, which come together in the presence of a minister, and then they are transformed or changed into newlyweds. The minister, of course, as we said, is unchanged. The minister is free to now go on and perform more ceremonies as long as there are brides and grooms available. Okay, so that's very much what happens in the case of enzymes. With enzymes, enzymes themselves are catalysts, right? They speed up a reaction or cause a reaction to take place, but they are not themselves used or changed by the reaction. Enzymes are made of protein. And when we talked about proteins in the earlier VodCast, we said that one of the important functions of proteins is to control chemical reactions. Well, enzymes are the way that we control chemical reactions. As far as I know, every chemical reaction that takes place in every cell of every living organism is controlled by an enzyme. Now, there's probably a couple of exceptions out there, but like I say, as far as I know, all chemical reactions are controlled by enzymes. Okay, now some specific terminology that is specific to enzymes. Enzymes have what we call an active site. Okay, it is the part of the enzyme where the reaction actually takes place. And the term for reactants that we use, specific when we're talking about enzymes, is we talk about a substrate molecule or molecules that attach themselves at that active site. Once those substrate molecules are attached at the active site, we now what we have we have what we call an enzyme substrate complex. Okay, so the active site is the point in the, or the area in the enzyme where the substrate or the reactants bind, and then they form an enzyme substrate complex. Okay, don't worry, I'm going to have lots of pictures later on to kind of show you what this is all about. Once the substrate reaction occurs, so once our substrate or our reactants react, now they basically float away, and our enzyme is free to be reused as long as there is more substrate molecules. Now, another very important thing to understand about enzymes is that they are specific. They are like what we use this analogy of a lock and a key quite often. A lock is only changed by one particular key, and substrates are only changed by one particular enzyme. Or probably, I guess the other way is true. Enzymes only work with specific substrate molecules. Okay, so let's see some pictures to kind of see what these things look like. First, I'm going to start off by showing you a model of an enzyme. Excuse me. Now, this is not a picture that I would expect you to draw into your notes. This, as you can see, this is an actual enzyme. This is an enzyme that breaks apart sucrose, which is table sugar, into its two component parts, which are glucose and fructose. So this is our enzyme, this red, gray, white, and blue structure. The red molecules, of course, are the oxygens, the grays are the carbons, the blues are the nitrogens, and the white ones are hydrogens. The green blob is our sucrose molecule, and this enzyme will break that apart into glucose and fructose monomers. Sucrose, of course, is a disaccharide. The active site is the notch over here on the side of the molecule, or the side of the enzyme, where our substrate fits in. Okay, so in this diagram, we're looking at the enzyme substrate complex, excuse me, which is formed by the enzyme combining with the sucrose substrate. Okay, this, as I said, is a fairly complex picture. We're going to show you some examples. I'm going to show you some examples with something that is a little simpler, a little easier to draw. Okay, first of all, we start off with our enzyme. In this diagram, you can draw in your notes, and I would expect you to do this. The notch over here on the side is our active site. So our enzyme is in green. This is a digestive enzyme, so we're going to be breaking apart this substrate molecule. So the purple thing, which will fit, as you can tell, into that particular active site, that purple gizmo will fit right in there. And when it does, that forms what we call our enzyme substrate complex. Okay, so now we have our enzyme substrate complex. The next thing that will happen is that the enzyme, since this is a digestive enzyme, it will break apart that substrate into the product molecules. As you can see here, now our products will leave the enzyme, and now our enzyme is free to find another substrate molecule and perform that reaction over and over and over again. Okay, to show you this in a little bit of a different animated form, okay, this green thing is, of course, it's our enzyme. It is going to interact with the purple thing, which is, yes, the substrate molecule. When those two things attach together, we have, yes, an enzyme substrate complex. The next thing that will happen is that the reaction will take place. So now you see our enzyme with our products. The products will float away. and now the enzyme can catalyze more reactions as long as it can find some substrate molecules. Okay, this is an example of a digestive enzyme. But enzymes can also put things together. That's how most of our biological molecules got put together in the first place, is by enzymes that can put things together. So here is an enzyme. And in this case, the substrate molecules, there is an orange one and a green one, which are slightly different shapes, and they're going to fit into the two active sites on this particular enzyme. Once that happens, we will have what we call an enzyme substrate complex. The next thing, of course, that happens is our enzyme binds those two substrates together, and we have now our product, which looks like this, which is the purple thing shown on the right side of your screen. Okay, now, so to show this in an animated form, we start off with, yes, the enzyme. Coming in from the right, we will have, yes, our substrate molecules. Once those three things are attached together, we now have, yes, an enzyme substrate complex. And the next thing that happens is that the chemical reaction takes place, and we now have our product. So the product now floats away, and the enzyme is free to go on and catalyze more reactions as long as there is substrate available. Okay, so now I want to show you a very specific enzyme that we're going to be working with in the lab this week. This is a kind of a fancy model, three-dimensional rotating model of amylase. Amylase is a digestive enzyme that's in our saliva. This is a different type of a model, but here we see the green thing is the big green blob is, of course, the amylase enzyme. The yellow kind of snaky looking thing is the is the starch molecule. Starch, as you recall, is a long chain of it's a polysaccharide which is formed by many monosaccharides, in this case glucose. Okay, so here is a different view, or a simpler model of the same thing. And the large gray blob, of course, is our amylase enzyme. Our substrate is starch, and the notch that you see in the middle there is the active site. Well, we had an earlier view of what starch looks like, and of course it is formed by these individual glucose molecules. Our amylase enzyme comes in, and it is a digestive enzyme, and its job, if you will, the reaction that it catalyzes is to break off these glucose molecules, individual glucose monomers, from the starch polymer. And so the way this particular enzyme works is by just working its way down the glucose chain, or I'm sorry, the starch chain, and as it goes along, it produces the glucose monomers by breaking them off the chain. So you see this happening, and this will happen again and again and again as long as there is starch substrate available. The amylase enzyme can do its job, which is to just break off those individual monomers. Now, as I said, we're going to be looking at this in some detail this week. I do encourage you to go back and view this VodCast again, because there is quite a lot of information in here, and it is really important that you become quite familiar with it as we go into the lab next week. So this has been Honors Biology for Desert Ridge High School. I'm Mr. Galladay, and I hope you have a great day.